your group, to your enthusiastic group of people. Yes, participants are requested to mute themselves during the duration of the talk. Yep, that's good. And also, if you want to ask a question during the middle of the talk, please go ahead and do that. It'll be fine. And then we can also ask other questions at the end. So could one person keep their video camera on so I can see a living human face on the screen alongside mine? Thank you very much. Okay, yeah, that's great. 2 of you is fine. All right, so you asked me to talk about this work, topological scaling laws and the statistical mechanics of evolution, and I'm happy to do so. It's one of the most surprising examples of how one can apply statistical mechanics and renormalization group ideas in biology. And usually when one applies physics in biology, what one is doing is typically looking at molecular biology or say the cell biology, mechanics of cells, looking at, say, molecular motors, maybe using physics methods to do advanced microscopy. But here I'm going to show you something very surprising, that one can actually study the statistical mechanics of the evolutionary process and learn something non-trivial about it using statistical mechanics. So this was a big surprise to us. And so let me first of all introduce my co-workers in this. So Carl Rose, who you will hear about in a few moments at the beginning of the talk, a very famous microbiologist, and actually somebody trained as a physicist. My student, Kalin Vatsigian, Patricia Geraldo, Hongyan Shi, Jiu Liu, Shi Xu. And all of these people did this work with me when I was still at the University of Illinois at Urbana-Champaign, and I moved from there 2 years ago to University of California, San Diego. So let me talk about the process of evolution. So most biologists would think that we understand evolutions, and they would say, well, we understand it. Darwin kind of explained it, and then the sort of mathematical theory of it was developed in the 1930s, and it gave rise to the field known as population genetics. But that's really not quite true. One of the things that we don't really understand is how it is that complexity increases without bound in living systems. So starting off from something that is abiotic, mainly just atoms and molecules. Please mute your microphone, Ankit. Thank you. Well, I'll mute it for you. You're muted. Okay. So life obviously started abiotically, but then evolution process led to advanced organisms like us and others. And the question is, why didn't it just stop at, say, something like the RNA world or some sea of self-reproducing molecules? What is behind this drive to ever-increasing complexity? And so that process is something that has not really been modeled until the work that I'm showing you here on the screen. I'm not going to be talking about this work, but let me just say something about what you're looking at here. So what you're seeing here is what I believe is the world's first simulation of digital life, organisms that can be said to be living in some sense, evolving in a computer. What you're looking at, the epilepsy-inducing video, is a picture, a representation of a system of trees, essentially trees, which are competing for photons. As they compete for photons, they compete for the photons in order to get light, in order to grow, but they're also evolving their genomes, and as their genomes evolve, their phenotype evolves as well. And so you're seeing here organisms that start off as very lacking in complexity at the lowest level, and as the simulation runs, they get more and more complex and larger, as you can see, and form clusters and so on. So why am I showing you this? Well, apart from being the first example of how a ecosystem has open-ended growth of complexity, this was derived using statistical mechanics. In fact, we know it has rigorously open-ended growth of complexity because we can apply ideas from scaling theory and renormalization group to prove that this is what is happening. Now, so if you're not a biologist, you might think that, well, what's happening is there's some mutations here that are gradually causing these organisms to evolve. But that's not correct. In fact, the mutations are not very powerful in terms of generating genetic novelty. And most of the novelty arises from two processes that we can prove are important in this model. One is gene duplication, a wholesale copying of a gene. And then the other is horizontal gene transfer, which is the transfer of genes from one organism to another to which it is not related. And these processes give rise to rapid evolution. They give rise to a network structure rather than a tree structure, and I'll talk about that in a few minutes. And the idea behind this model was developed by thinking about the inverse energy cascade in two-dimensional fluid turbulence. So this is what is happening here is you're getting a cascade of complexity, as we called it, which is analogous to the energy or entropy cascades in turbulence. So that's all I'm going to say about this. I will return to this topic at the end of the talk and maybe in questions afterwards. So what I want to spend the next 45 minutes talking about is I want to show you what is the phase diagram of large-scale evolution. And so basically I'm going to show you the history of life on Earth. And then I'm going to talk about phylogenetic trees, which are the genomic footprint of evolution. And in ways that I'm going to make very precise, these are in fact the Feynman diagrams of evolution. And I'm going to show you how one can understand, how one can quantify the structure of evolution from these trees that we can observe and measure, and deduce non-trivial scaling rules, which I'll explain what they are, and show you how we can make a model for this using principles from statistical mechanics. At the end of the talk, I'll talk a little bit about some philosophy here, which I hope you'll find interesting and perhaps even inspiring, to explain what I think are the reasons why we need to understand better the evolutionary process from a fundamental perspective. Is there a question? Okay, if there's no questions, then please mute your microphones. So somebody has their microphone unmuted. Okay, so this is the New York Times, November the 3rd, 1977. And the person you see here is my friend, who's now deceased, unfortunately, Carl Woese, in his office at the University of Illinois. And why is he there? Why is a microbiologist who was originally a physicist on the front page of the New York Times? Well, he had just published a paper in the Proceedings of the National Academy of Sciences, and this paper, as the New York Times put it, discovered a form of life that predates higher organisms. Actually, that's not quite true. In fact, he discovered not a form that predates higher organisms, but actually that coexists with them, but had previously been unobserved. And the way that he did this was he decided that the best way to try to map out the history of life on Earth was to look at molecular sequences extracted from organisms, and this was started in 1967, long before the current era of genome sequencing. But his work was a sort of the basis for genome sequencing, and his idea was take all these molecular sequences, choose a particular part of the ribosome which is highly conserved. The ribosome is a machine that makes proteins in every cell of every living creature that we know of, and by looking at the genetic sequence of this, one can see basically it's basically very conserved, but the small differences allow you to map out the history of the evolutionary process. And what was known before his work was that there are two kinds of cells. There are eukaryotes and bacteria. So eukaryotes are cells which have a nucleus, and bacteria are cells that do not. That's the usual picture. But what he discovered, in fact, that some of these cells that look like bacteria cells are these things called archaea, which are as genetically different from the eukaryotes as the bacteria are. And here you can see the map. So this is where you are. Virtually everything else on this tree is microbial. Rose discovered this entire branch of the tree of life, the archaea. They're usually found in very extreme environments. They can also be found in your gut or in soil. So they're quite common. They're not just some pathological species or a curiosity. They're very important for the Earth's biogeochemical cycles, for example. Now, when you construct the molecular sequences and compare them and construct these trees, and I'll say a little bit more about these trees in a few minutes, what you find is that there's a root of these trees, and the root of these trees is what is known as the last universal common ancestor, or maybe the last universal common ancestral community or community. And so it's called the last universal common ancestor because this everything that is alive today was descended from this organisms or community of organisms. Now, there's three problems in early evolution that I want to draw attention to. So the first thing is, let's start with number 2 here. This last universal common ancestor that I just showed you, we can date it because the phylogenetic tree, the lengths on it correspond to DNA single nucleotide polymorphisms. So we can date it, and we also have geological evidence from stromatolites and so on. And so we know that this last universal common ancestor is about 3.8 billion years ago. And that last universal common ancestor we can detect using this method, this molecular method that Rose invented, and that means that those organisms had pretty much the same machinery in their cells as we do today. So the basic architecture of the cell was already around about 3.8 billion years ago. Well, here's the puzzle. The Earth is about 4.6 billion years old. And so that means that life started from nothing to getting to essentially the complexity of the modern cell in less than 1 billion years. And that's amazing because after that 3.8 billion years, there's been hardly any development of the cellular architecture. And so the question is, why did the cell evolve so rapidly initially and then afterwards so relatively slowly? Then there's a second question is, we know that there is only a single canonical genetic code. When I talk about genetic code, I mean the map from triplets of nucleotide bases on DNA to the amino acids, the 20 amino acids that make up all of life's proteins. And so that's what the genetic code is. And it's basically the same for all organisms, apart from very minor variants that I'm not going to really talk about. So that's the second question. Why is there only one genetic code? Why not many? And lastly, when you examine the genetic code, what you find is it's close to the theoretical optimum for minimizing errors in gene expression. And so there is some kind of variational principle, and we'd like to understand why. So here is the genetic code, just to make sure that everybody knows what I mean. So you have nucleotide bases depending on whether it's RNA or DNA, UCANG or TCANG. And the way you read this is you say, let's look at this point here in the code table. So I read this, and then I read this, and then I read the second column here. So you can see this amino acid is phenylalanine, and this is U, and U, but there's also U, and C as well. So, and the same thing for all of these other amino acids, and stop means stop codon. So that's the canonical genetic code, which is basically universal, and it's also optimal in minimizing the effects of errors. So what I mean by minimizing the effects of errors? Well, you can have errors in two ways. One is errors in translation. One is errors which arise due to point mutations. So you might say, well, okay, that's great. If you were to design a genetic code, you would say, what I'd like to be able to do is say, make the code such that if I have some kind of error due to a substitution or a translation error, the amino acid that I get has very similar properties to the one that I should have got, so that the proteins fold and the organism still survives. That's what you would like to be able to do. So how can we explore that issue when we only have one genetic code available to us? So the answer is do Monte Carlo simulation. So you take the genetic code that I just showed you, now you permute the labels of the amino acids. So I have 20 factorial, which is about 10 to the 18 possible synthetic codes that I can generate. And so I can sample from these codes. And once I sample from these codes, what I can do is I can create a measure that tells me how good this particular code would be in minimizing errors compared to the original one. I'm not going to go into the details of that, but the basic idea is to take a measure of polarity that was measured by Carl Woese in the middle 1960s. And this measure measures the balance between hydrophobicity and hydrophilicity of these amino acids. And then as you swap them around in the genetic code, you take the sum of the squares of the differences of their polar number. And from that, you can generate a number and you can compare it with a canonical genetic code. So it's not important how that is done. There's many non-unique ways to do that, but they all give you the same answer. So now what you do is you measure the code property, this particular score, this polarity score.
ये recording आएगा तो भेजो मेरे को.
What? Pardon? Was there a question?
No, I guess someone mistakenly unmuted. Please carry on.
Okay, so you look at the probability distribution of this code property, and there's two possible things that could happen. So you might expect that, as Francis Crick did in the 1960s, that the genetic code is a frozen accident. So a frozen accident means the following. And it's an accident because we're all descended from some organism, whatever it happened to be, just historically that was an accident from which we're descended. And the genetic code is frozen because if I start changing the genetic code, then the proteins I get will stop making sense. They'll be the wrong proteins, the wrong amino acids. And so therefore, as I read the genome, I'll make the wrong proteins and the organism will die. So Francis Crick predicted that the genetic code would be a frozen accident. So if that were true, then if I generated genetic codes at random, get this sort of distribution I've shown you here in the blue, and then I looked at where the actual genetic code is, let's say here, it should be part of what I would get from a random distribution. The other possibility is that it's not like that at all. So here is my bell-shaped curve telling you about the random distribution of scores for genetic codes chosen at random. And if the real genetic code is down here, and smaller is better in this way of scoring things, then that would tell you that the code is not a frozen accident and something special must have happened. So when you do the actual calculation, as was first done in the middle 90s, what you find is that here's your bell-shaped curve for the scores based on random codes, and then here is the actual value for the real actual genetic code. And so what you see is that the genetic code is very far away from what you would expect at random, and so it tells you that the code is pretty near optimal in minimizing errors. In this paper, it was called the genetic code is 1 in a million. Today we know that it's much more like 100 or 100 million or even more than that. So a very highly optimized. So how did it get that way? I'm not going to answer that in this seminar, but if you're interested, you can look at this paper, which showed how the genetic code evolved by collective mechanisms which involve horizontal gene transfer. And I'm not going to go through it, but I'm just going to tell you that it was not point mutations that caused the code to evolve, it was horizontal gene transfer. Horizontal gene transfer, as I said, is a process by which an organism has genes that are transferred to another organism, either by picking up DNA from the environment, or by transferring plasmids between them, or by having a viral-mediated generalized transduction, where a virus breaks into a bacterial cell and basically destroys the cell, reproduces itself, but takes some of the cell DNA with it and then infects another cell and transfers the DNA to that. So we know that microbes today can do this, and they do this with antibiotic resistance genes. But what happens when they all do it, in terms of the evolution of life, it turns out that they enormously accelerate it. because the passage of genes in a population now becomes something which is a network rather than a tree. So here are simulations that we did showing you the genetic code quality over time. This is in red what happens when you have no horizontal gene transfer, just regular vertical Darwinian evolution. Genes are given to your descendants in the usual way. And you can see that what happens is the code evolves a little bit and then gets stuck in some local minimum and then freezes. And so this is the frozen accident theory coming out, and you can see that the results are indistinguishable from what you would get at random. But if you now switch on this horizontal gene transfer process, the organisms evolve very rapidly, and the code quality improves to a point where it is much better than what you would get at random, and that's shown.
Sorry to interrupt, but this graph is from the simulation.
This graph is from a simulation, yes.
Okay. Yeah.
So, and it's not specific to the simulation details, it's generic. So horizontal gene transfer leads to optimality, and it also leads to uniqueness in the code that you get. So what you learn from that is that the phase diagram of life looks something like this. I showed you already a phylogenetic tree, and there was the last universal common ancestor. That time was 3.8 billion years ago. But there was an earlier period of time when life started, underwent a period of very rapid evolution through a network effect, which sometimes is known as the progenote. And this collective network phase of life makes a transition to the vertical evolution as shown over here. And so one of these processes is fast because of network effects. One of them is slow because it's on a tree. Okay, so that's the history of life. Now I want to talk about the tree phase of life. So here again is a phylogenetic tree. And as I told you before, what we do is we can observe extant organisms, which are the edge of this phylogenetic tree, and we observe them by looking at the sequencing data for certain genes that code for a part of the ribosome. And then the inside of the tree, the organisms that you see here, which represent the evolutionary process, are derived by comparing the relatedness of sequencing data and deducing what must have happened at earlier times. And there's many different ways to do that, and we understand that very well. And this is what you get basically. So phylogenetic trees represent the trace of the evolutionary process, and to a physics audience, I would say that they are the Feynman diagrams of evolution. And I mean that quite literally, as you'll see. So now I want to talk about what we can learn by examining these Feynman diagrams of evolution. So now I'm going to talk about work that was done with Chi Xu, who was a graduate student with me, and Jiru Liu, who was an undergraduate at Illinois and is now a graduate student at Stanford. So here is a phylogenetic tree, and you can see it's a very complicated structure. If I take the blue part of it and blow that up, you would say it looks pretty much like the original tree. So you might say, well, phylogenetic trees are some kind of fractal or something like that. But we can do better than that. We can make a quantitative description of their structure. So let's do that. So there's two aspects of a phylogenetic tree. So one is its topology, which is showing you here. So the topology doesn't change if I change the length of these various edges. It just tells you the connectivity of these nodes. And these nodes in the phylogenetic tree tell you about new species. And how you define species, you probably know from eukaryotic organisms. How you define species for bacteria or archaea is a little bit more technical and more complicated, but you can do it, and so we're not going to talk too much about that. So you can look at the topology of this tree and admire its structure like this. You can talk about a metric measure of trees by looking at these edge lengths here. These edge lengths represent numbers of mutations. And so you can see there's a sort of molecular clock ticking away, and the evolutionary process is happening in this time of the molecular clock. Okay, so I'm going to start off by talking about the topology. So if you don't know anything about topology, don't worry. I'll explain it to you right now. It's not too important that you understand exactly the details of what I'm showing you. I just want you to understand that one can construct two measures of topology, and you can do that through a kind of recursive algorithm. So here is a sketch of a phylogenetic tree, and we're going to assign two numbers to each of these nodes, the nodes representing speciation events. So the first number is called A, it's the node number, and what we're going to do is we're going to give a 1 to every node. So each node here has a 1 which represents itself, and then another number for each of the nodes descended from it. So this particular node here just goes to the present time. So time is increasing in this direction. This is where you are now. This is going backwards in time. So this one here is just a 1 because it's just itself. This one has a 1 because it's just itself. This one here has the number 3 because it has itself plus 1 plus 1. So you get 3. And then this one here has itself, which is 1 plus 3 plus 3, and that gives you 7. So that's the node number. Now there's another number which is, does somebody have a question?
Yes, sir.
Yes.
Sir, is it similar to something that we use for weighing the nodes as in graph theory, but we are not actually measuring the nodes they are arising from?
It is a construction that if you've seen graph theory, there are things like this in graph theory, yes.
So we are not considering the one they are arising from, means we are not actually considering the
No,
one
it's just recursive for the node and the ones descended from it. But maybe what you're thinking of is this one. So this is the cumulative node number. So in this one, what I do is I take the node number for itself, and then what I do is I add I add its node number plus the node numbers of the two nodes underneath it. So this is a recursive algorithm that sums all the nodes below it. So this number I'm calling C. So for this node here, it is 3 for itself plus 1 plus 1, and that gives you 5. Okay, this one here is 7 plus 5 plus 5, and that gives you 17. And you can do that recursively. So the question we want to ask is, I've got these two numbers, and the question is, what is the functional dependence of C versus A? In other words, how does the cumulative node number depend on A? Now, why is that an interesting question? Well, suppose I have a symmetric tree, or a balanced tree, as we might call it, like the one I showed you before. So it looks like this. If I ask, because this is a recursive algorithm, you can compute this exactly, and you can find that C of A asymptotically goes like A log A. So that's nice. If you have a completely unbalanced tree, where at every bifurcation, one branch goes down to the present time, and the other node goes to some other time and then splits, so maximally unbalanced in the way shown over here, you can show that then this goes like A squared. Now, if you take all the genomes that you can lay your hands on, and we did this in about 2012, somewhere around then. Harada et al. Did this around 2008 independently. I'm showing you Harada et al.'s data here, and plot C versus A. What you find is it's a log, it's a straight line. It's basically a power law with an exponent which is about 1.4. I wouldn't put too much faith in the second digit here, but something like this. So we're seeing that we have a strange power law intermediate between these two extremes. And so the question we would like to understand is why is that true? Now, there's a second metric I want to talk about, which is about the edge length distribution. Now, this one is more involved, and I guarantee you that you won't capture it in your minds when I explain it to you. So I'll show it to you and you'll sort of say, okay, I kind of get the idea. What's important is that you see the scaling laws that I'll show you from the data in a few minutes. So what you do is you look at clades. So you look at a clade as a group of, is a node here which has tips that go down to the present time. So this is one which has 2, for example. So you let I, the set of I and K, be all nodes that define a clade with K tips. So this clade has 2 tips, this one has 3 tips, and so on and so forth. And then you let Li be the edge length between I and its branching point. And then you sum up all of those edge lengths. So let me give you an example. So here's the tree I showed you before. So let's suppose I set K equal to 1. So now I'm looking at nodes that in one step go down to the present time. So I add up the edge length distributions of the edge lengths of those. So I add up 10 plus 4 plus 4 plus 2 plus 2 plus 2 plus 3, and I get 25. Now I look at the ones with K equals 2. So there's two of those. There's this one and there's this one. These have got two tips. This one has got one, this one has got two. That's the edge length from the branching point. And so you get 3. And I can do the same thing for k equals 3, 4, 5, and so on. Okay, so once you do that, then you can ask, well, what do I expect that distribution to look like? So let's suppose that I had a randomly branching model. I call it a so-called Yule process. What you would find you can show is that S will scale like k to the minus 2. On the other hand, that's if you think of such a randomly branching model, the number of organisms is going to be growing exponentially, which is not realistic. So then you can say, well, let's assume that there is a constraint on the size of the community, and then that means that some of the ancestors have to be common. And when you work that out, it's called a thing called a Kingman coalescent. What you get is a scaling law that goes as k to the minus 1. Now, my colleague at Illinois, James O'Dwyer, who actually was also a theoretical physicist when he was up to the time he did his PhD, became an ecologist. He showed that if I look at microbial communities, various ones chosen in these various different environments, this scales at k to the minus alpha, with alpha being somewhere between 1.3 and 1.7. So what I've shown you are two stylized facts that describe the topological and metric structure of the large scale of evolution. And so now we want to understand where do these scaling laws come from. So we want to understand what is their origin, and we want to know what, if anything, do they tell us about living systems. Well, if you want to understand non-trivial power laws in biology, it behooves us to understand non-trivial power laws in physics. So let's take a look at critical phenomena, such as in say a magnet. So here, if you've read my book, you'll know that here's magnetization of a magnet, number of spins pointing up versus number pointing down versus temperature. And below TC, there's a phase transition to an ordered state with a magnetization that scales the way that I've shown you here. This is in zero external field. If I look at the phase diagram in the external field and temperature plane, it looks something like this. So here is H equals 0. This is the phase transition shown over here. And there's two stylized facts that come from these pictures. So here we see that the magnetization scales like T minus TC to the power beta. That's shown over here as T tends to TC. And then if I sit on the critical isotherm, so I sit at this critical temperature here, and I ask, how does the magnetization of the magnet depend on the external magnetic field? Now, normally you'd say it'd be linear response theory, so M would be proportional to H, and that would be Curie's law. But that breaks down at the critical point, and in fact, you get a non-integer exponent, which is called 1 over delta. And what Widom and Kadanoff discovered in the 60s was these results and others follow from the idea that the magnetization, ostensibly a function of two variables, T minus TC and the external field, actually is really just a function of this one composite variable over here. And here is the proof of that. So here is the magnetization from 5 different magnetic materials. These are real experimental data. Magnetization plotted in the way that theory predicts, temperature plotted in the way that theory predicts. Here are all the data, and they all collapse onto one universal curve. And the solid line that goes through here is the renormalization group prediction for this, and this has no adjustable parameters. So this is really quite fantastic. This shows you that this, the model, the Heisenberg or Ising model of a magnet, is giving you a very precise prediction in agreement with experiment, a triumph of theory. But it's not really true. This actually is a model of a model of a model of a model of a model. And what you're looking at here is a model which starts off at quantum chemistry, then goes into electronic structure, then say a quantum spin model, then a classical spin model, and then finally Landau theory, and then eventually a renormalization group theory. So you're making a series of non-systematic approximations. None of these approximations can be derived systematically. There's no small parameter. And these are just a sequence of minimal models. And the renormalization group tells you in retrospect why it is that this completely illegitimate process of simplification actually works and gives you precise results. Now, the origin of the non-trivial power laws can also be understood in a fairly simple way. So if you look at the two-point correlation function of the magnetization in a magnet. So this is essentially the power spectrum, if you will, of the Fourier transform of the magnetization in space. I evaluated as a function of wave number and temperature at the critical temperature. If you look in my book, you'll see that it goes as k to the -2 plus eta, where eta is some anomalous critical exponent. And you might say, well, how can this possibly be true? Because if you do the dimensional analysis of the statistical mechanics, which you can read in chapter 7 of my book, what you find is that the correlation function has units of length squared, while the wave number has units of inverse length. So you would naively expect that this g of k and t, c, would scale as k to the -2, and it would be impossible for it to have an anomalous exponent. But what's happening is scale interference. So what happens is, even though the correlation length of the system is called arbitrarily large because you're at or very close to the critical temperature, the lattice spacing, which I called A here, actually enters into the expression for the correlation function. And it enters in this singular way, a to the power eta, in such a way that it cancels out this eta over here, and so dimensional analysis is restored. So what this tells you then is that even though the correlation length of the system has diverged to infinity, the system still remembers small scale details of the lattice spacing. And this is called scale interference. It defies common sense, but that's the only way to understand how you can get anomalous scaling exponents in physics. And so the question we want to talk about now is there scale interference in evolution? So here's the evolutionary process that I've shown you before, and let's have a look then at how scaling laws could arise. So here are the nodes of the phylogenetic tree, and they represent ecological processes. So you might have a speciation event that happens, and then the speciation creates some new species, but that new species might then immediately be eaten up by others in the environment, and so it doesn't persist and enter the evolutionary record. So there's a whole range of evolutionary processes by which a new species can eventually spread and fill an ecosystem, and eventually, as geneticists call it, go to fixation. So these nodes of the phylogenetic tree hide the ecological processes. Now, the evolutionary processes happen at long time scales, and the ecological ones happen at short time scales. So what we're doing when we are drawing a phylogenetic tree is we're setting the ratio of the ecological timescale over the evolutionary timescale to be 0. Just like you might have thought that in a critical phenomenon, you could set the correlation length of the system or the lattice spacing to the correlation length would also be very small and could be set equal to 0. But what I showed you is that it can't. And so now we're going to see, is this also invalid in an ecological system? Well, the quick answer is yes, it is invalid. We know examples of how evolution and ecology can happen on the same timescale. What happens is, typically I have some population of organisms which interact. They create, they interact with each other through their metabolism. They create waste products that create new biochemical niches for other organisms. Those organisms then alter the environment. The environment then provides selection on the population structure that we have here, and this whole process begins again. And this is something that's known in ecology as rapid evolution, and you can see it very simply in experiments on phage and bacteria, in rotifers and algae, and I'm not going to go into this, but the actual population level signatures of evolutionary processes happening in real time can be seen by looking at anomalous phase relationships in predator-prey cycles, for example. If you're interested in that, you can read my paper with Hong-Yin Shi in this place over here. So so evolution can affect the entire trajectory of an ecosystem, and so that tells you that there's a feedback between ecology and evolution, and so therefore this timescale separation may break down. Now, since you're physicists, I'll show this slide. If I want to think about these ecological interactions, they actually literally are Feynman diagrams. So, for example, if I have a predator and a prey, I can write down in terms of quantum field theory ways that you can take an ecosystem to find annihilation and creation operators that create and destroy organisms in the environment. I can say A as a predator, B as a prey, and I can say when a predator meets a prey, so A plus B, there's some probability the predator eats the prey and then uses the energy obtained to make a baby predator. So A plus B goes to A plus A. These are interactions, and they are quantized because individuals in a population are coming units of one, just like photons, electrons, protons, neutrons, gluons, and also persons. Okay, organisms are quantized. So you can write these processes down using coherent state path integration, and the Feynman diagrams that correspond to this look something like this. And for each of these Feynman diagrams, you can see the particle picture, the wave particle duality that you can see over here. And if you...
Yeah, like this the quantization, we can draw the analogy between QFT and evolution, right?
It's not an analogy, it's mathematically correct, it's mathematically exact.
Okay.
So what you do is this isn't just an analogy. You can take a classical stochastic process, write it down as a master equation, and then you can write down a sort of Hilbert space that defines the Fock space of the system, and define inner products and quantum operators on those spaces. And so basically the matrix element tells you about the probability rather than the matrix element telling you about the probability amplitude as it does in quantum mechanics. But each of these things can be written down as a path integral. And so this is an exact mapping of the classical stochastic process into the language of path integrals and quantum field theory.
Okay.
And I'll show you where you can read about it. So here is vertex renormalization, as you may know from quantum chromodynamics. Here is vertex renormalization in evolutionary ecology. So here is a phylogenetic tree, as I showed you before. Here are these nodes. These nodes, as I told you, represent ecological processes. If I do the vertex expansion of those nodes, then as I'm showing you here, you can see the Feynman diagrams that represent the vertex operators for these nodes. And if you want to read about it, you can look at my papers, but they're quite complicated. The simplest paper I would recommend would be this paper by Uwe Teuber in the 2012 Journal of Physics A. So this is a nice kind of review article about this formalism. Okay, so now let's go back to the biology. So what you would normally say in evolution is that the environment selects for the fittest phenotype of the organisms. So the way that the organisms evolve in time depends on the organism, but it also depends on the environment. Now, normally what you would say is, well, the environment itself just changes by geological processes, perhaps, something like that. So, but the idea of niche construction is that there is a feedback between the organisms and they change the environment as well. So in some sense, the way the environment changes involves both the organisms and the environment, and the organisms, they change, evolve based on the organisms and the environment as well. And so evolution now becomes a collective behavior. And so biologists have had this idea for some time. They call it niche construction, and it's the feedback loop that I was showing you a few minutes ago. Now, the most famous example of this is about 2.6 billion years ago, the Great Oxygenation Event, where the cyanobacteria proliferated on Earth. They produced oxygen as metabolic waste. They poisoned all the anaerobic organisms that lived at the time. So the oxygen accumulated in the atmosphere. So they literally poisoned the planet, just like we're doing through global climate change. And then when the oxygen level was high enough, it could support high energy organisms like us. Without the cyanobacteria, organisms like us that have very high energy would never have evolved. And why do you need high energy? Well, if you think about what oxygen is, it's basically rocket fuel. And we are essentially powered by rocket fuel and electricity. And that's due to the phase transition engineered by microbes 2.6 billion years ago. Okay, so So
One
yeah,
question. Yeah, can you please go back to the slide of that environmental niche construction? Yeah, so that O, that stands for organisms. I mean that DO.
yes.
Okay. So what that
Mm.
I mean that equation tells that first.
It is just a metaphorical way of saying that the evolution of organisms is coupled with the other organisms, that's ecology, and also the environment itself, which is the other organism, and the same thing is true for the environment too.
Okay. So
So
it is function of
both these
organism and environment.
both of these things are intertwined, whereas the classical textbook assumption is that the environment is not changed by the organism. So you wouldn't have the O over here. So that's the new thing, having the organisms involved in changing the environment. Okay, so let's make a simple, minimal mathematical model of it. Just like the Ising model is a minimal model of a magnet, and there is no justification for the simplifications made in deriving the Ising model. So we're going to make a model that also is very simple, unjustified, but justified in spirit, and see if we can extract universal behavior from it. So we take these nodes in a phylogenetic tree and we associate with them three numbers: the available niche, the speciation rate, and the extinction probability. And the physics of this is that an organism with a large niche value has a large number of possible ways to adapt to the environment. Let's suppose, we know that diversification happens in evolution after there's been mass extinctions. So when there's many available niches to be colonized, then there's a lot of diversification, therefore a lot of speciation. So we'll make a model of it that goes like this. We take the niche numbers of say these ones here, descend from what is above, and then there's a fluctuation on that, which is a geometrical Brownian motion shown over here with a standard deviation, which I'll call sigma. That's inheritance. Speciation says that the more niches there are, the more likely are to speciate. So the speciation rate R will be an increasing function of n, the number of niches available. And of course, if n is negative, which is unphysical, then it will just be some constant, which may end up being 0. Okay, so that's that. And then, of course, there's only a finite amount of resources available. And so we have to bound the growth of the tree by including extinction. Okay, so those are three processes. We can generate phylogenetic trees from those. And here I'm showing you those phylogenetic trees. So this is C over A as a function of A. And I'm varying here the strength of the fluctuation, the magnitude of the fluctuations in the inheritance rule. And what you can see happening is that if that is 0, then I get these data over here, and that actually scales like A log A, which is a balanced tree. As I increase this, what is happening is the tree crosses over to asymptoting to this straight line here with an exponent which is about 1.5. Okay, so similarly, if I look at the edge length distribution, I get the same thing, that as I increase this, the standard deviation, I get a scaling law for the edge length distribution. So what I've shown you is that this very simple model reproduces both the topology and the edge length distribution that you see in real phylogenetic trees. Okay, so that's that. Now, are these scaling laws really anomalous parallel scaling? I'm going to skip over this, but you may want to know where does the singularity occur from? And it occurs because this scaling law here, that the speciation rate as an increasing function of n, is only valid for n greater than 0. N less than 0 is not allowed, so there's a singularity at this boundary. And the higher the standard deviation is, the higher chance you have to reach the boundary, and so you generate more asymmetry. And then you can do various crossover and scaling analyses and show that this is really what's going on. Okay, so what I've shown you is that these non-trivial power laws arise from niche construction and the interplay between ecological processes and evolutionary processes. So that's the first thing. And the second thing is, what do they tell us about living systems? Well, it tells us that evolution is not just mutations and horizontal gene transfer. One must take into account the ecological dynamics that leads to genetic fixation, even on timescales of billions of years. Competition, predation, range expansion, metabolic cross-feeding, and so on and so forth, all leave an indelible footprint on the evolutionary process. Okay, so I want to spend just a couple of minutes talking about some sort of slightly philosophical ideas, but tell you what I really want to explain to you is why the study of evolution is something that is interesting for physicists in and of itself, even if you don't care much about biology. So I went to a conference virtually a couple of years ago during COVID called Is There New Physics in Living Matter? And that's what I want to talk about for the next few minutes. So one question that one might ask is, do we learn new physics when we study biological physics, or is it just insanely complicated soft material science? In other words, why should physicists care? Are there any universal phenomena in biology? Do they reveal anything important? And do we miss something by not understanding them? And I'm going to show you that there are universal phenomena. They do reveal something important. And actually, we really suffer by not understanding these universal phenomena. So I'm going to whiz through this. I'll probably spend about 5 to 7 minutes on this, and then I'll conclude. So the idea of building something to do with universal biology is a kind of dream, which is very difficult to conceptualize. So let's talk about something that we do understand, which is universal computation. So here I'm showing you two computers. The one on the left is like our laptops that we have, all of us have on our desks. The one on the right is a piece of clockwork springs and cogwheels that was built and were designed and partially built by Babbage in the 19th century. And both of these things are computers. Okay, one of them works, one of them would have worked if it had been finished. Both of these things are built out of different materials. This is a shiny chunk of glass and plastic and silicon. This is a bunch of cogwheel springs and levers, but they both have something in common. And what they have in common is that they are an instantiation of an abstract concept, which is a Turing machine, a universal Turing machine with a von Neumann architecture. And that can be instantiated in many different ways. So here you are, here's another Turing machine with a von Neumann architecture. Here's the ones I already showed you. This is a John von Neumann in front of the MANIAC, one of the world's first computers, built at the Institute for Advanced Study in Princeton, primarily for the purpose of not just exploring computation, but also doing the very complicated hydrodynamics calculations for designing nuclear weapons. Now, when the earliest physicists who went into biology, like Schrödinger and Delbruck and others, they hoped that they would find new laws of physics, particularly quantum mechanics. Today, not so much, perhaps. People like me tend to think that the real new physical laws are the laws about emergence and the existence of life itself. How does matter self-organize hierarchically to create replicating and evolvable structures? How do molecules come to life? That's what we'd like to really understand, not so much the quantum mechanics. And when we talk about understanding in physics, we usually mean two things. First of all, we understand why something happens at all. And then secondly, we understand how to calculate how it does happen in a particular physical system. So we understand. So we understand, say, electromagnetism, and then we also understand if we have an electrical circuit, we can compute its properties. Now, I want to claim that if we want to understand biology, we need to understand from a physicist's perspective what these things have that is unique. And the thing that is unique about biological systems is that they can reprogram themselves. So what do I mean by that? So on the left here is Niels Baricelli. So Niels Baricelli was a visitor at the Institute for Advanced Study when von Neumann was building the first computers there. And after the engineers had finished their bomb design during the day, he went in and used the computer and built the world's first computer viruses. And this picture was given to me by George Dyson. And it's a picture, it's a complicated picture of these computer viruses competing for memory in the core of the computer that I showed you. And Niels Baricelli observed that they underwent diversification, and they competed and they evolved and things like this. And so the main thing about these computer viruses is that their computer code was the thing that they acted on. So when they 8 other organisms and so on, what they were doing was modifying each other's code. So if you like, the program is the data. I know data is plural, but it's better like this. The program is the data is the program. The data has it means two things. It means the organisms, and it also means the program itself. So these are living systems, or living systems in a computer, are able to reprogram themselves, and that's how they're able to respond to perturbations by creating new functionality. And the selection pressure of evolving digital organisms in the computer, like the ones I showed you at the beginning of the talk, causes them to evolve. And this aspect of living systems or self-reprogramming systems is very different from physical systems. Physical systems have a Hamiltonian, and that's it till the end of time. But biological systems, or evolvable systems, can change their Hamiltonian. And the way they change their Hamiltonian also can change. And the way they change the change of the Hamiltonian also changes, and so on ad infinitum. And so what happens is, I believe, that the future state of an evolvable system is inherently unpredictable, and I suspect that it is Gödel undecidable. And I think it's Gödel undecidable because biological systems can evolve new functionality, and once they have some kind of novel mutation or novel horizontal gene transfer that gives them new capabilities, they have new phase space, they have new degrees of freedom. And once they have new degrees of freedom, the phase space in which they were previously living has now expanded, might even have changed its cardinality. And so this feature means that they're unpredictable, but it also has some very dramatic consequence, and that means it's very difficult to control an evolving system. Okay, so are there things that are universal in biology? Well, there are, but I'm going to skip them for the interest of time. Is universality useful? Yes, it is. No, it isn't. I'll skip all of that. I want to get to the last slides, which is what do we miss by not understanding universal phenomena. So since you're physicists, hopefully this will make some sense to you. I want to talk about superconductivity. And the thing I want to talk about with superconductivity are these seminal discoveries in superconductivity, which actually, at least 3 of them got Nobel Prizes: the discovery of superconductivity, the zero resistance, the discovery of the Nigel Goldenfeld effect, which is the expulsion of magnetic field, discovery of the Abrikosov vortex lattice, and then lastly, the discovery of the Josephson effects. And both of these things were predicted by theory. Now the question is, why did it take so long to make these discoveries? Superconductivity was discovered in 1911. Why did it take 50 years to discover all the phenomena associated with superconductivity? And it's even more strange because if you take my class on emergent states of matter, for example, or any other class that you're taking on advanced condensed matter physics, you will know that you can derive all these things in a couple of lines of algebra from the ideas of off-diagonal long-range order and a charge condensate coupled to an abelian gauge field. So basically, a very simple quantum symmetry and field theory explains all of these things, explains why superconductivity exists in the first place and all these phenomena. And it explains it not just in material science, but also in things like quark stars and so on and so forth. So the reason is that physicists until I'd say the 1960s or 1970s were not very cognizant of the idea of levels of description. And the idea of levels of description was something that the renormalization group really brought to the fore. So let's end by talking about levels of description. I'm going to compare superconductivity and biology. So here's levels of description in superconductivity. You could have quantum chemistry and material science, for example. You could have, say, the BCS theory, Bardeen-Cooper-Schrieffer theory, for interacting Cooper pairs. You could have the Ginzburg-Landau theory for off-diagonal long-range order plus an electromagnetic gauge field. So those are 3 different levels of description that you might apply to, say, a superconducting material. In biology, you have other levels, for example, the level of atoms and molecules. You might have the level of description of DNA, where you can think of DNA as being random coils and helices. They have some kind of stiffness, and so there's some elasticity theory for DNA. And then at the highest level, you might have the thing that we've been talking about today, the universal dynamics of evolving systems. These different levels of description answer different questions. So in superconductivity, this level of description tells you, for example, how do specific materials instantiate the Bardeen-Cooper-Schrieffer mechanism for the origin of superconductivity. So you'd want to know that in some particular material if you wanted to, say, understand its critical temperature. The BCS theory for interacting Cooper pairs tells you what is the basic mechanism in weakly coupled Cooper pair superconductors for the formation of electron composite bosons to form a Cooper pair. And the Ginzburg-Landau theory, an order of diagonal long-range order plus electromagnetic gauge field, that level of description tells you why the phenomenon of superconductivity exists in the first place and all of the phenomena that arise from that. Now, in biology, you have the level of description of atoms and molecules, elasticity theory for DNA, dynamics of evolving systems, and they describe different things. This one describes, for example, how do specific biopolymers interact and fold, undergo template-directed synthesis, for example. This level of description applies to understanding the basic functional cellular processes. And this level of description can tell you why the phenomenon of life exists and what it does, its general response. These different levels of description have different levels of universality. So this level of description for superconductors tells you about specific materials, for example. This level of description tells you only about weak coupling superconductors, and this level of description applies to all superconductors. In biology, this level of description applies to very specific biopolymers. You're knowing the atomic and molecular sequence of a particular biopolymer. This level of description tells you about, say, the phase transitions in liquid-liquid intracellular complexes. It tells you about the physics of subcellular components. And this level of description tells you about all of life. Now, why do we need this universal level of description? Well, in superconductivity, we have this level of description which applies to all superconductors. And if you don't have it, as we didn't for the first 50 years of superconductivity, we didn't have a way to predict the response of a superconductor to an electromagnetic field. We couldn't predict the Meissner effect, the vortex lattice, and Josephson effects, and so on and so forth. In biology, not having dynamics of evolving systems, which applies to all of life, means that we cannot predict the response to selective perturbations. Now, in superconductivity, by regarding superconductors as collections of atoms, we are missing the emergent laws that act at the large system scale and govern the large scale response to the electromagnetic fields. And we know how to solve this problem. In biology, by not having that level of description, we don't know how to predict what happens when you perturb a living system. How might you perturb a living system? Well, if you've got a pathogenic bacterial infection, you'll apply antibiotics. If you've got bugs that are eating your crops, you'd apply an insecticide. If you've got weeds that are growing in your plant land, you'd apply herbicides to kill the weeds. If you've got cancer, you would apply chemotherapy agents to try to kill the cancer tumors. If you don't have those things, what happens is you develop antibiotic resistance, you develop insecticide resistance, you develop herbicide resistance, and you develop chemotherapy resistance. Every time we intervene in complex biological systems, treating them as engineered systems, as physical systems, the system reroutes itself around the control mechanism that we've tried to apply, and the control mechanism that we've tried to apply no longer works. So by regarding biology as complicated physical systems, we're missing the emergent laws that act at the system scale and govern the large-scale response to control perturbations. And we don't know how to solve this problem yet. It's important because we have a world health crisis with the emergence of antibiotic resistance, particularly in India, I might add. We have the same thing with the emergence of pandemics such as SARS-CoV-2 and so on. And so this is a very fundamental feature of living systems which impact our ability to construct healthcare. And we don't know how to solve that problem. And to solve it, we need new mathematics and new physics. So do we learn new physics when we study biological systems? Absolutely. There are universal phenomena, and what do we miss by not understanding them? We miss the ability to respond and control biological systems. So my message is this one, which was first enunciated by Stan Ulam, paraphrasing John F. Kennedy, the US president from the 1960s. Ask not what physics can do for biology, ask what biology can do for physics. Meaning, don't just say, can I use physics to build a better microscope or a better computer simulation? Instead, ask, how does biology expand the scope of physics? As I think, as I hope, that I've shown you, biology raises questions in dynamical systems and statistical mechanics that we don't encounter in just purely physical systems. And so by doing so, by studying biology, we expand the scope of physics. And with that, I will leave you and I'll be happy to take any questions.
Thank you, Professor. I think if anybody has any questions, they can just unmute and ask.
So unmute and put on your cameras and feel free to ask any other questions.
Yes, Professor. I wanted to ask a question. Yeah, I'm a sophomore student, and I have recently been working upon the Ising model, 2D Ising model simulation of it, and I got to know something about ergodicity. And whenever I tweak the model, 2D Ising model, Metropolis algorithm, I tweak it. Somehow it it doesn't take all of its possible states. It become non-ergodic. Is there right now I don't have certain topics that I have not covered. That's why I don't know more about ergodicity. How can I make my algo ergodic? And is it that if we want to model a whole of biology, or like a simplistic model, if we want to know about it, there has to be some part of computation also, some great questions that are still unanswered, many great problems like PNP problem, what Clay Institute has put up on its like algorithms algorithms behind everything. These algorithms are only working, right? And we cannot make things work until unless we don't know about more of things, how algorithm connects.
Okay, so the question, so yeah, I heard what you were saying. I'm not quite sure. So the question is, how do we make algorithms for describing different processes? Is that what you're asking?
Yeah, such that it would give results as we suppose it to be giving. 2D Ising model, sometimes I tweak it and it doesn't form a clump that I wanted it to make at certain temperatures.
So what could be happening is that you know, when you modify your Ising model, you're changing the critical temperature. So it may have broken ergodicity, and you may be in a broken ergodic phase. So you may think that you're in the paramagnetic phase where it should be ergodic, but you're in the ferromagnetic phase and it's actually got broken ergodicity, but you didn't realize that because the transition temperature shifted on you. So where you thought you were in the paramagnetic phase, the change you made did that. So it could be that, or it could be that the particular modification you're making has some error in it, I suppose.
Non-local.
Non-locality definitely can cause these sorts of things. So so yeah, there isn't a universal prescription that says, here's how you make this thing. I mean, physicists have intuition about how you should construct minimal models, and like in my research group and other people's, people know how to build simple models and can guess what are the universal behavior. So for example, people a lot of physicists are studying these days active matter. They're studying flocking. They're studying how, say, birds or fish will form a flock. And maybe 30 years ago, 25, 30 years ago, various statistical physicists went and tried to guess simple mathematical models of that, which they did, and showed that they could explain some of the phenomena that you see in flocking and so on. So it really is the intuitive process by which physicists make guesses about how to make models of nature, that has not been replaced. But then you can use algorithms and maybe renormalization group theory or other things to sort of test those guesses and compare them with experiment. So that's the way people do it. So anyway, I don't know if I answered your question. But
Yeah,
I'm not quite sure what the question.
like in this grand quest of finding the model of whole biology, wouldn't it be
there's no, I don't think there's such a thing as a model of all biology, because the thing is that you know what you have to understand that if you can understand universality, it is good for answering certain questions, but it's lousy for answering others. So let's suppose I'm a condensed matter physicist or a material scientist, and so I say, you know, I want a grand model that describes all superconductors. So I have one for you. And then my experimentalist friend says, well, that's great. So what is the critical temperature of this particular high temperature superconductor? I can't calculate that. So he says, well, what good is your universal knowledge? So sometimes you do want to know very specific things. That's a completely valid question. And those will tell you quantitative things about the superconductor. But they won't, but having that knowledge won't necessarily tell you about you know, things like, you know, the Josephson effect that you would otherwise never have known existed. So there's different types of theory for different types of questions. So it's not really true to say that there's a sort of universal equation for all of biology. I don't think that's a good way to say it.
Yeah, thank you.
Okay, so I see some other questions. So Adish Kilikar, yeah.
Hello, Professor. Yeah. Thank you for your talk. I really enjoyed it. And I'm working on disease models, particularly on like dengue and livestock disease and all of that. So right now our approach is using mechanistic models to atoms and so on. And these kind of models are actually used for national vaccination strategies also. So I was interested in your last point where you mentioned about how, you know, regarding biology as complicated physical systems can may not be able to capture the large scale dynamics.
You're breaking up, so I really can't hear you. Maybe turn off your video camera. Perhaps you'll have more bandwidth for the sound.
Kind of model sort of audible. Sorry. Yeah.
Yeah, none of your question came through, I'm afraid.
Was I not audible?
No.
Okay, I'll repeat the question. So
Yeah.
yeah. So I work on mechanistic approaches to study diseases, and these models are used for national vaccination strategies also, like how to do the interventions and so on. So since you mentioned that these treating biology as like physical systems, we are missing emergent properties. So what would be an alternate approach to understand these systems for these kind of interventions? Because we don't have any other option. So what would be an approach? For example, in disease models, we miss out serotypes and we have to make some compromise, some simplifying assumptions and trade-off.
Yeah, no, I understand your question. I had the same experience because I worked on modeling COVID-2, on COVID as well, during the from 2020, early 2020 until late 2021, and you know, it was very clear that there was a failure of simple epidemic models in predicting things like the you know, even the next 4 weeks of a pandemic, and providing information to public health authorities, providing things like, say, when the epidemic will peak. There's a failure in trying to understand why there are plateaus, why there are waves of pandemics, and so on. And so I was doing public health epidemiology for the governor of the state of Illinois. And I set up with other people at Illinois a PCR testing for the entire university population, testing 50,000 people twice a week using saliva rather than sticking things up your nose, and we use that to control the pandemic. Now, there were a lot of things that went wrong with that, and not just with us, but with other people as well. It worked, but one of the things is that you can't predict things outside of your original outcome space. So we had a situation that was actually became quite famous. It was in the New York Times, and Twitter went crazy about it, and so on. We modeled how the students would behave, and you know, the other people in the university, but particularly the students who had a lot of social activity. We included parties and things like this, and yet the rate of COVID, the first week or so of the winter semesters, for fall semester in 2020, there was a slightly higher positivity than we expected. Still a factor of 10 lower than that experienced at other universities, but still a little bit higher than we expected. And the reason for that was, we discovered, was that students were actually not obeying the law. They were, even though they knew they had COVID, they still went to parties and held parties and deliberately infected other people. And so that was something that we didn't foresee. Nobody foresaw that. And it's just something that you can't take into account. So that's an example of how, well, once we knew that, of course, we could monitor for that and calibrate it and so on. But it was something that was a new thing that happened, like the novelty in evolution that I told you about. Another example is, we discovered that if you include the social activity of people stochastically alongside the mechanistic model of the progression of the disease in the population, that gave you much better predictions for the course of the epidemic, and when it peaked, and how high it would peak, and so on. So those are examples of really sort of collective population effects that at the beginning, just by focusing mechanistically on the disease and not focusing on the population who are infected by the disease and understanding their dynamics and the couple dynamics, you make inaccurate predictions. And it's very much like the niche construction I talked about in the talk. So did I answer your question, or at least peripherally address it?
Yeah, that answered the question, but I'm still confused about, I mean, there are so many specificities we miss when we try to generalize, and I still don't understand what, you know, are there any rule of thumbs that we generally follow when we course train these models, or so on?
Yes. Well, yeah, thanks for reiterating the question. So, you know, it became very obvious to me early on in the pandemic that just writing down, you know, compartmental models of an epidemic and ignoring the population was the wrong thing to do. So the first thing we did was we studied, you know, what is the thing that we're leaving out, which was the population. And the most important feature of the population is it's heterogeneous. So the heterogeneity in the population makes a big difference to the course of the disease, but that doesn't feed back into the disease. So then we included the social activity feeding back into the disease progression as well. And once you have that in there, then you have a much more realistic a much more realistic prediction of the features of the pandemic. Just like, say, Ginsburg-Landau model of a superconductor tells you in general about all the features that can happen. It won't tell you about, you know, the specific critical temperature of a particular superconductor, but it will tell you all the things that happen. And so the kind of models that we made eventually, by looking at what we had ignored and putting those things in, then did restore a much better qualitative agreement, and then we could calibrate those models to data and make forward projections. So there isn't a simple equation. You have to look at what you're doing and think about it and see what it is that you're missing out, and then see whether there are collective effects that could be important.
Yeah, thank you for your answer. I got it. One more follow-up question is,
if you're interested in that, there was a paper I wrote in eLife. It was published in 2021, and the editor of it was Mark Lipsitch, who's a professor at Harvard and is the director for the CDC's Center for Disease Forecasting. And he wrote a little editorial about the paper, and then you can read the paper itself.
thank you. I'll check that out. Also, one more question was, now because of the emergence of AI and machine learning, a lot of people are using it for predicting the case incidence. But how do you view this, this new trend of using these kind of models to predict something that is inherently very unpredictable?
Well, I think it's a good idea to use AI to try to predict, to try to extract from the experimental data features that you can't model quantitatively. So you might be able to follow the disease progression by looking at the cases and the hospitalizations and the fatalities and so on and so forth. But that still won't tell you very much about the mechanisms of spread, about the heterogeneity in the population and so on. And so it is possible that machine learning could add those extra ingredients into a model that is that uses the sort of mathematics of epidemiology and the way I said to provide forecasts, but enhanced by including these other factors that we don't really know how to quantify. So I think that could be successful, but I haven't seen anything that convinces me that that's been done yet. But it's something that quite a few of us are thinking about actively at the moment.
So any precautions like we should take when we do all of these approaches to model diseases?
What precautions should you take? Don't hang up your brain. Think. You have to use judgment. Don't rely on some complicated computer simulation or some complicated AI or some arbitrary set of equations that somebody wrote down. You still have to use your brain.
Yeah, that would be, yeah. Thank you for your answer. Thank you for your time.
Yeah, good luck with your studies on disease progression.
Yes, thank you.
Hello, sir. Can I ask a question?
Go ahead, whoever's talking.
Yes. So thank you for this wonderful talk, firstly. And my question is, when you were describing about how we had that last common ancestor. So before that, you said that evolution basically happened, like it evolved very fast, and we came to the stage of a very well-developed cell, and those cellular processes were, developed by that time in a matter of say 1 billion years, right?
Yeah.
What I wanted to ask is, why did that horizontal? So you said that horizontal gene transfer was the main feature in that, the main mechanism for that to happen. So why did that transition into tree-based evolution? Like, what caused that? Do we understand that?
Yeah, we do. So there's a paper I wrote in 2017 that talks about that. So the mechanism for horizontal gene transfer. So let me just say a little bit about that since you asked. So can you wait? Adish, can you please mute? Yeah, I'm going to mute you. Let me try to answer your question. So gene duplication, I mentioned at the beginning, you accidentally make 2 copies of a gene. One of them carries on doing what it was supposed to do. The other one, now it can be mutated, and it doesn't matter. so it won't adversely affect the organism. If that one turns out to be a better version of this, then it can actually help the organism provide some positive feature. So that's how you can evolve by gene duplication. And horizontal gene transfer is the import of a whole set of genes, or maybe just one gene, into a population, into an organism that it gives it a completely new functionality. It would be like if there was a gene for doing 4 minus epsilon expansions in quantum field theory, and I just threw out that gene and gave it to you, and then suddenly you knew how to do it, rather than having to read my book or any other book on quantum field theory. That's how effective it is. Now, how did that happen in early life? Well, early life had a lot of endosymbiosis. That means the organisms were very floppy and porous, and they can collide and engulf one another, and they can exchange genetic material in that way. And when we build these trees of life, what we're actually looking at is the history of particular genes. We define species based on the translational machinery, the machinery that reads the mRNA and turns it into proteins. So because you have to do that in order to have life, at least life as we know it, we define the species based on whether they have the same translational machinery, and in particular, the same 16S small subunit of the ribosome. So that's how that process happened. Lots of organisms crashing into each other, swapping genes, and eventually what happened was the machinery became sufficiently advanced that two things happened. One was that any new version of the machinery that you got was actually identical to what you already had, so it didn't do any good. And the other was what you had was so complex that any new machinery that you might transfer into it would just not work. So the analogy that I sometimes use for that,
like saturated, it basically got saturated, you're saying, right?
Kind of saturated. Think of it like this. Think about the evolution of automobiles. Okay, so early automobiles, like in the 1890s or something like that, early 1900s, they were very rickety things. Their top speed was like 15 miles an hour or something like that, and they were built by inventors using bicycle parts and very simple engines and things like this. Okay, and let's suppose I drove my car over to your house, and maybe something went wrong with one of the wheels. I would say, well, how am I going to get home? And you would just say, well, here, I've got a spare bicycle here, take the wheel off that and put it on your car and off you go. And so that you could do that, in 1900. Okay. Now imagine that you've got a modern car, like, say a Toyota or something like that. Well, obviously, if something goes wrong with the wheel, putting a bicycle wheel on it is not going to work. You won't be able to do that. You can't even get it repaired. You have to go to a special garage where they will have a special computer so that they can interface with the modern day Toyota and fix it and so on. So as the machine gets more and more complex, it becomes less and less tolerant of having new things added to it. And so the combination of those two processes meant that eventually horizontal gene transfer of the core machinery of the cell became ineffective. So you still have horizontal gene transfer happening today. Mostly today, it's happening in your stomach right now. The bacteria that live there are transferring antibiotic resistance genes, and that's how antibiotic resistance emerges. But the core cellular machinery, the stuff that makes the cells work, that is now basically fixed. And so that's how that transition occurred.
So in a way, we can say that as we evolved, basically, the higher order organisms, they are less flexible. So that is why it's the bacteria that develops, say, the antibiotic resistance. And we say if we are given some other blood group type also, we would not be able to even sustain that, right? So I think it's fair to say that although we, as complexity is increasing, our flexibility or our maybe adaptability is decreasing, and maybe that's why archaea can live in such extreme conditions. Is that all connected?
It is. You're right about that. And if you read our 2006 paper, we talk about this as tolerance of ambiguity. But that's exactly what is happening. Now, archaea, the fact that they live in hot temperatures, or can do, doesn't mean because they evolved earlier. They didn't evolve earlier. All of these things evolved more or less the same time. So it's not that they were older and so therefore they could tolerate hotter environment. You know, the thing that lived in those environments had particular characteristics, you know, different kinds of cell membranes, different kinds of processes going on inside the cell, and that's how they were able to do it. Okay, great question. Yeah, thanks.
Hello.
Yep. Hello.
Yeah. Hi.
Hi there.
Yeah. So again, great talk. So you mentioned when you were talking about scaling of magnetization with external field and temperature, and you say that it works well, but it's a system built upon system built upon system, and renormalization group validates the scaling. And now, and then you move on and you do the same thing for ecological dynamics. So now, is there like, again, this is also like these ecological dynamics, that is also very coarse grained. So now, is there something that validates it, like what RG did for this Ising model?
Yeah, no. Well, there should be, and there could be, but I haven't done it. But there could be. Some of these, you know, when you say RG validates it, what RG specifically tells you is that there are certain fixed points of the renormalization group transformations which are defined by things like symmetry and dimensionality, and then all the other things, coupling constants and so on, you can show are irrelevant at a particular fixed point. Now, for something non-equilibrium and something as complicated as these evolutionary processes, it's not obvious how one could do that. But it's not obvious that there's any small parameters. So in some sense, you'd be like a renormalization group before people had tools like, say, the epsilon expansion or the 1/n expansion. So in principle, one could do that, but it hasn't been done at the present time. So if you want to go ahead and do it, please, I invite you.
Like on a related note to this, so you talked before about behavior and how difficult it is to model behavior. And I think this also makes biological systems very different from physical systems, is that physical systems are universal and they have particular length scales. Now, biological systems, in addition to having these length scales, they have these evolutionary behavior. So it's like a certain bacteria also has to perform behavior, but it has to perform in a, let's say, an overdamped limit. And for, let's say, for us humans, we also have to perform the same behaviors. And these behaviors can be nonlinear, and even they might not follow the same, like from physics, many laws come from symmetry, right? We have conservation of momentum, conservation of angular momentum, and these will not be the same for biological systems. So like, how would we even come up with such laws?
Right. So the thing is that one has to realize that the laws that we care about are the laws at the emergent scale and not the laws at the microscopic scale. So when we're talking about things in biology, the degrees of freedom that we're using are, you know, say if you're an ecologist, you're talking about, you know, population density of some creatures and things like this. Okay, well, by far that's way many orders of magnitude above, you know, atoms and molecules and so on. So nobody cares about, say, the symmetries of the atomic Hamiltonian and things like this. So what you care about are emergent symmetries, such as, for example, in turbulence. Okay, you take a fluid, it has all sorts of symmetries, you know, translation invariance, blah, 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 conservation of momentum, angular momentum, whatever. Now you go and put your fingers in it and stir it up and make it turbulent. Now you get new symmetries that arise in the turbulent fluid, right? Scale invariant symmetries and multifractal scaling and various things like this. And those symmetries, you can't really see them from the original microscopic ones. So if you want to understand, say, something as complicated as turbulence, my viewpoint is the worst place to start is the Navier-Stokes equations. Just like trying to understand the symmetries, the emergent symmetries of, say, biology, the worst place to start would be the Schrödinger equation. Okay, you'd be crazy to do that. So you have to choose the right level of description. And when you choose the right level of description, there are then emergent symmetries that correspond to that level of description. And so, we don't know we don't know much about how you classify such things. We do know that even in the simplest models of systems that are in a steady state, but in a non-equilibrium steady state, we do know that the universality classes are much more complicated than they are for equilibrium statistical mechanics, for example. So that's kind of where the field is at the moment, trying to understand how do you go from, you know, what we saw happen in the very simple case where you have Gibbs distribution, equilibrium stat mech, to non-equilibrium steady states where you have all sorts of other distributions which are generally unknown and emergent symmetries, and then trying to apply that to even more complex systems. Well, that whole field is still in its infancy. So people would like to know the answer to your question.
Yeah, no, because like for Ising model, you have spins, and an emergence of that is that like this curve of magnetization that you get. And in biology, something similar could be, let's say, a polarity of a cell, and eventually you get an organogenesis. So you have different like structures forming. And I can even wrap my head around that, but still, when behavior comes into action, so you have like animals, they are competing. They're competing for food. They're competing for mating. They're competing for various resources. Now, how this could come under the same framework?
Well, I can give you an example of that. So if I go back to the work I did on epidemic modeling. So what we said was, okay, we know that humans are heterogeneous. They're heterogeneous in their response, in the susceptibility to the SARS-CoV-2 virus. So that's one thing. They're also heterogeneous in terms of their social activity. And social activity, of course, is something that drives pandemics. So that's a kind of relevant variable. So some people, you know, just like to sit at home and solve chess puzzles and do physics problems and read textbooks on normalization group theory, and other people like to go out to the nightclub every night and dance and have a good time. And so people have different steady state levels of social activity, but they also have transient activity, meaning that during a pandemic, well, you say, well, boy, it's, you know, I don't want to get this terrible illness, so I'm going to just sort of hide in my basement and just watch TV for the next few weeks and hope it blows over, kind of thing. And then eventually I'll return back to my steady state, and I'll go out to the nightclubs, or I'll go to the library and study the normalization group theory. So people have different transient responses and different heterogeneous steady states. And what we found was just including a simple model of that was enough to already explain a large disparity between what you see in simple models of an epidemic and what you see in real data. So I think the only thing you can do is really try to use your intuition to try to say, well, this is something that we can add, and this will make a qualitative difference because it's something that's obviously being left out, and you can see it's important. Now, then you might say, well, okay, fine, now let's put in some extra feature, like the fact that people like to go out on Saturday night, especially because in the weekday they have to go to their job and, you know, lectures at the university and stuff like this. So basically everybody parties just on Saturday night. Okay, well, that's certainly true, but is that going to make a qualitative difference to the behavior of the system? Probably not. So that's an example of an effect which is just, well, tuning some coupling constants, the strength of social interactions, whereas the first thing I told you about is actually including the social interactions in the first place, expanding the state space of your description of the system. And so that's how you can use your intuition to see empirically what things are relevant and what things are irrelevant. Changing, say, the description of social activity to being, you know, it only happens on Saturday night. Okay, that's just like, say, changing the electron-phonon coupling constant in the superconductor, and that will just change the critical temperature from 23 degrees Kelvin to 25 degrees Kelvin. So that's things like what?
Being more mechanistic, like moving toward more mechanistic basis than phenomenology.
Well, it's not a question of being more mechanistic. It's a question of some things give you quantitative answers, more quantitative answers, improve the accuracy of a prediction that you can already make qualitatively. Okay, so if I add particular details of, say, the electron phonon coupling constant in a superconductor, that makes my prediction of the critical temperature to be more accurate. But it doesn't change the fact that I can predict the critical temperature in the first place. That just comes from the BCS theory. Similarly, if I have an epidemic, if I take into account social activity, that makes a huge change in your description of the epidemic. It tells you that the epidemic does not go up and then come down as epidemic models predict. It tells you that you get these consistent waves and plateaus and things like this. It predicts that. A qualitatively different, qualitatively new feature of pandemics. Now, if you then say, okay, now social activity only occurs on a Saturday night, does it change that anymore? No, it doesn't. But it might make a slight change to the frequency of the oscillations or something like that.
So like picking the right level of description, as you said before.
Taking the right level of description and then adding more and more realism just changes the accuracy of the prediction, but doesn't change the qualitative nature of the prediction.
Okay. Yeah.
If you're interested in this question, I wrote there's a volume, there's a book coming out called, I think it's called something like 50 Years of Renormalization Group Theory, and it's a tribute to Michael Fisher, who was a statistical physicist who perhaps you've heard of, and my article for that is on Archive. It's called There's Always Room in the Middle or something like that. So it's the latest paper I published on Archive, but it talks a lot about these minimal models and what they can and cannot do.
Okay. Thank you so much.
Sure. Ashmi.
My question is that how does horizontal gene transfer lead to the decrease in average amino acid distance between the codons, as we could see from the graph?
Yeah, thank you for the question. So what I want you to imagine happening is this. If you think of it's not quite right to think of it like this, but it's a good analogy. If you think about evolution as being some kind of optimization process. So you can imagine I've got some landscape that looks like this, which has some global minimum. And I'm starting at some place and I'm doing global descent. So I'm doing gradient descent on this landscape. So if I'm just following local rules, I will just descend locally, and then eventually I'll hit some local minimum, and then I'll get stuck there. Now, and that's what mutations do. Now, to get around that, what you can do are global rules or stochastic gradient descent or other names for it, where you can have a rule that tells you can hop around the landscape, and by hopping around and going down, eventually you're guaranteed to reach the global minimum. And so what horizontal gene transfer is a non-local move in your landscape of genotypes. So I've got a landscape of genotypes, I've got some fitness on top of that landscape. And then what is happening is horizontal gene transfer is a non-local move that suddenly brings in some new genetic material. And so now it swaps you around, moves you around on this landscape in a way that is non-local. And that's how it enables you to find the global minimum. And so that's how you end up finding the best genetic code and the unique one.
Thank you. And one more question, that is, the genetic code is close to the theoretical optimum in minimizing errors in genetic expression. This was mentioned in one of the slides. So what is meant by this? Please, can you explain?
Cool. So the genetic code certainly is very good at minimizing errors, but I'm not claiming that it is exactly the optimal code, meaning you might be able to find a code that does slightly better. But again, it's not really a very good statement, that, because if you measure, how do you measure how good a genetic code is at minimizing errors? Well, there's no unique way to do that. So there's many different ways that people propose, and they all more or less, when you simulate them, give you the same result. So the result is robust under the way in which you define optimization. But remember, in evolution, it's not really true that it has to optimize something. Well, evolution can stop when it's good enough that it is able to overcome any selection pressure. So it doesn't have to be optimal. There's no variational principle that guarantees it has to be optimal. So that's why I say it is close to optimal.
Okay, thank you.
Sure.
Hello, sir.
Hang on, Ashmin Cow has that hand up. Was that?
Yeah. So am I audible?
You're audible, a little bit muffled, but yes.
Okay. So I just want to thank you for this interesting talk. So the thing is, this was my first talk about using physics to interpret biological systems. Earlier, I was never able to connect like something as abstract as physics to something reading like biology. So this talk has given me new perspectives and valuable insights. So thank you for giving me that new kind of perspective.
Great.
Thank you.
Thanks for saying so. And actually, I realized let me just share a slide. I didn't show this slide, but I meant to. Let me share my screen. So thanks for your question, or thanks for your comment. And if you are interested in this, there's two papers I wrote that you might enjoy reading. So this one is called "Life is Physics: Evolution as a Collective Phenomenon Far from Equilibrium," and this is the reference. But you know, if you just go to my webpage. On my webpage, you'll find my publications, and on my publications, you'll find this, and all the PDF files are there, so you can just download them, even if you don't have a subscription to the journal. So this is.
I'll do that.
Yeah, so that's this paper, and then this one is, I think maybe one of the last ones I wrote about the statistical mechanics of early life. There's other ones as well.
Yeah, thank you.
Okay, yeah. So anybody wants to just take a screenshot, and I'll hide these.
Hello, sir.
Hello.
It was a nice talk. I have a doubt. Like you asked questions initially during the discussions that why we have only one genetic code for like all other organisms. Can you just answer the question, like why one?
The answer is that if you assume initially that there are multiple genetic codes, which is certainly what you would have expected, you can show that they will compete with each other, and eventually one genetic code will become dominant.
Okay,
And,
so it is just like competition, and or like accidentally we got only one genetic code.
It's competition between genetic codes, not competitions between organisms. So I'll give you an analogy. If you ask what is the most common computer operating system on the planet, well, it's probably Android, which is in billions of smartphones. But apart from that, on desktop computers or laptops, it's probably Windows. And you might say, well, why is that? Is Windows better than the Apple operating system or Linux? Well, no, not particularly, but it's fine. But the reason is that initially there were a lot of people using Windows operating system, and Microsoft made sure that that was happening. And then when people were developers, so they would write computer programs just for the Windows operating system rather than the Apple one, because they would be able to sell more copies. And so the Windows operating system became dominant because you had access to a greater pool of innovation, of code innovation, that was accessible to you. So you had the Windows operating system, you could find computer programs that would do anything you wanted. If you had the Apple one, that wasn't the case. And in fact, when phones were, smartphones were invented with iPhone back in 2007, 2008. The big selling point was actually the App Store. You could get apps that developers wrote for the Apple phone, which was one of the first ones, and that's how it became very, very well known very quickly. Google then created Android or bought Android or whatever, and then developed the Android ecosystem. And so in many areas that now superseded iPhone, again, for the same reason. There's many apps that you can get. So the basic idea then is that communities of organisms that share the same code base can swap innovations by, say, horizontal gene transfer. And so the organisms with the access to the greatest pool of genetic innovation will be the ones that ultimately outcompete the other ones, even though there is no direct selection on codes themselves. And that's all
So,
explained
like,
in my
is there any evidence of other life forms which uses other codes, or is it just a theory?
There are other life forms that use slightly different codes, but they're not very major changes to the codes. As an example, the mitochondria in your cells they were due to an early endosymbiosis, and that organism that was engulfed eventually gave up most of its protein making ability to the nucleus of the host cell. But the mitochondrial genetic code is somewhat different from the nuclear genetic code. And there's other organisms that have slight variations depending on their environment, but they're not major ones.
Okay, thank you, sir.
Sure. So I'm going to have to go in like 2 minutes.
Yeah, so then maybe we can end the talk here.
I think the n by dt is negative now.
Yeah,
it's
15 now.
It's late in India, so
so yeah, thank you for this wonderful talk, especially this interesting subject of the interface between biology and physics. Yeah, thank you.
Thank you. I hope you enjoyed it, and I hope you all enjoy studying physics and do interesting things with it.
Thank you.
Okay, thanks for inviting me. It's been a pleasure to talk to you. Thanks for your interest, and thanks for your really well thought out, great questions. Really enjoyed your questions. They were very thoughtful and very, very smart.
Thank you, Professor Goldenfeld. Thank you.
And if you want to apply to graduate school in the US, the University of California, San Diego is a very good graduate school, particularly in biophysics.
Thanks. Surely.
We'll bring you on that.